for the next 3 days. Today, we are going to talk about statistical analysis in MSstats, specifically for quantitative experiments with TMT labeling. So I hope that this will be of interest to you. If you happen to join us for the first time, let me just review the logistics. Please use the chat box at the bottom for things such as, hi, how are you? We can't hear. But for all questions related to science, please use Q&A, because then we can manage the questions easier, and we can also save the questions and post them on our docs document so that you can review the answers later on. Okay, so then we will go ahead and get started. So the plan for today is that first, we will have the introduction to the MSstats ecosystem by Devon Koller. So Devon is a PhD student in the Curie College of Computer Sciences, and he is one of the lead developers of MSstats. He has done a lot of work, in particular with MSstats PTM, and also he is the lead developer of MSstats Shiny. So we will hear more about this today. Then I will speak about the statistical methods specifically implemented in MSstatsTMT, which is one of the packages in the MSstats ecosystem. If you're joining us for the first time, my name is Olga Vitek. I'm a faculty in the Curie College of Computer Sciences at Northeastern, and our lab is the main developer of MSstats. And let me right away introduce Ting Huang, who will then do some hands-on analysis talking about how we actually work with TMT-labeled experiments. So Ting holds a PhD in computer science from Northeastern University. And as part of her research, Ting has developed a lot of the statistical methods, which we will discuss today as part of MSstatsTMT. Very recently, Ting graduated, and she is now a research scientist at SEER, continuing to work in quantitative mass spectrometry-based proteomics. So we're very excited to have Devon and Ting with us today. So let us go ahead and get started. So first, we will do the overview of the MSstats ecosystem. Thanks so much. So Devon, you can go ahead.
Awesome. Okay, hopefully everyone can see my screen. Yeah, like Olga mentioned, I'm going to go over the MSstats ecosystem today, just go over what packages are available, how you can use them, and then their kind of current status and what we're developing and what you can expect in the future. So first, I'm just going to go over the team so you can kind of get a face to everybody who's developing everything. I've been developing some of the PTM work, MSstats Lip and Shiny, like Olga mentioned. Mateus is now the lead developer of the base MSstats. Melanie has developed some of the Galaxy implementations. And then Mina and Ting are both former members of the lab. Mina worked a lot on the base MSstats, and like Olga mentioned, Ting worked a lot on the TMT side. So if you have any questions or post in the Google group or anything, it's probably going to be me or Matt or Olga that are really looking at it, just so you know who is actually going to be there to answer any questions that you might have. Okay, so I think Olga showed this slide a bit yesterday. This is kind of the high overview of all of the different packages and how they work. So I wanted to go through it real quick and just kind of show you what is going on, because it can kind of be a lot. So at the top, you can see all of the different signal processing tools that we currently have converters for. So you can see like Skyline, MaxQuant, Philosopher. We don't have converters for every package. So for example, our newer packages like MSstats PTM and Lip only have a couple of converters. So we are actively developing the converters, but MSstats and MSstatsTMT should have most of these converters available for you. All of these converters are contained in this new package that we created called MSstats Convert. You don't actually need to interact with this package. It's more of a backend for all of the other MSstats packages. But we have it here just so you kind of know like how everything works. And if you're looking at the code, if you see MSstats Convert, it contains all the code for these converters. MSstats Convert is called by basically everything, but let's just focus on MSstats and MSstatsTMT. Base MSstats is used for significance analysis of DDA, DIA, SRM. It includes, like we went over yesterday, it includes visualizations, quality control, sample size, statistical analysis, everything that we went over yesterday. MSstatsTMT also uses MSstats as the backend, but it's specifically for tandem mass tag quantification. We have two UI tools for MSstats and MSstatsTMT, Galaxy, which Melanie developed, and also MSstats Shiny, which we went over yesterday. So only MSstats and MSstatsTMT are currently available in the UI tools, but MSstatsPTM is also being integrated into Shiny right now. MSstatsPTM uses some of the backend from MSstats, MSstatsTMT, and MSstatsConvert, so it's kind of built on everything. It supports statistical characterization of PTMs, and we can remove some of the confounding with the global protein if you have an unmodified run that we can use as well. MSstatsLib is built off of MSstatsPTM, so it can also remove some confounding as well. And then you'll see kind of not really with any arrows pointed to it, MSstatsLODB, which is for estimation of limit of blank and limit of detection. It has some packages for calculation or functions for calculation and visualization, but it doesn't really rely on anything, so it's kind of there on the side. But it is on Bioconductor, so you can use it if you would like to. And lastly, on this slide, we have the public data repository Massive Quant. So we have integrated MSstats into Massive Quant, so you can see if you have like an experiment, if there's some sort of quantitative analysis, you can go in and see not only the R code for the entire workflow, but also kind of the different steps. So you can see like, okay, here is the MSstats required data format. Here's the quantification, summarization files, and then here's our differential analysis files. So it's kind of just a way to tie everything together and to make everything reproducible and be able to give out the package and data to everybody. So that's a general overview of all of the different packages. MSstatsLib is available. It's available on Bioconductor right now. And I'll kind of go through each package individually and show kind of their status. MSstatsLib is our newest package, so I think it was just put on Bioconductor in last October, but it's still in active development, so we just released a kind of new update just a couple weeks ago for it. Okay, so first let's go through MSstats. This is a bit okay, so let's just start with the downloads. So we over the past year, we've seen 621 downloads per month for MSstats. The current version is version 4.41, and this includes some refactored code, which was actually implemented previously, but I just want to go over it because it did increase the speed and memory consumption of the package, or decrease the memory consumption, increase the speed. How we did this was replaced a lot of the tidyverse or dplyr functionality with base R and data table functionalities. And we also moved a lot of the core conversion functionalities to MSstatsConvert, which helped us be able to just share those conversion functions between packages. Currently, we're developing some new functionality to analyze very large data sets that don't fit into memory. So we've been running into some experiments with maybe like 50 gigabyte plus data files that like basically you can't load into R. So Mateus has been working on trying to just a functionality where you don't actually have to load it all into memory and then process it, so that we can actually handle those big data files and do analysis on them. So that should be coming along shortly. It is available on Bioconductor and GitHub, and I've included these slides in the Word document, so you can click on them. There's links there for all of this stuff. Okay, so next we have MSstatsTMT. MSstatsTMT has 234 downloads per month over the past year. Its new version is version 2.4. And in this version, we include that refactored code. So MSstatsTMT is based on MSstats, so it has MSstats Hall refactored code to increase the speed, and it also has itself has been refactored, so it should be a lot faster in this version. In addition, Ting just released in the new version a new model to account for time series designs. So time series designs was already built into MSStats modeling, but now we can also do it in MSstatsTMT, which improves our variance estimation of those designs. And I think I shared before, but the way you actually specify what kind of design it is in your annotation file. So there's a specific way you need to set up the bioreplicates and everything. So I can share that kind of vignette again so everyone can see exactly how to do that. Again, available on Bioconductor and GitHub, so you can install it either way. The development version is on GitHub and the live version is on Bioconductor. Okay, so next is MSstatsPTM. MSstatsPTM is a newer package. It's gone through a couple different iterations. So over the past year, it's had 109 downloads per month. Version 2.0 is on Bioconductor now. Version 2.0 contains a couple different things. So previously, we had a package called MSstats PTM TMT, TMT PTM, which included PTM data for or PTM functionality for TMT experiments. But what we did is just combine that functionality with the base MSstats PTM. So now that other package PTM TMT has been discontinued, and now everything's just in this core function, which includes functionality for both label free and TMT. So you'll see if you actually use the package, you'll just have to indicate whether your experiment's a TMT or label free experiment. We also replaced the backend with the refactored MSstats code. So before it was kind of on its own didn't have like integration with the rest of the family of packages. So now because we replaced the backend with the MSstats code, we improved the speed and reduced some of the memory usage. So the package should function a lot better as well. This package was developed with as a collaboration with Genentech. And we also have it is available on Bioconductor and GitHub. On the development side for this package, I think I mentioned before, but we are actively working on integrating the package into MSstats Shiny. One of the challenges with doing that is that the package, since it's pretty new, we don't have as many converters as we do for base MSstats and MSstatsTMT. So we are actively trying to build more converters into the function. So right now, the main converters we have are for MaxQuant and Progenesis. And it's doubly hard because we also have to find converters for TMT and different types of experiments. So if you do have an experiment that we don't currently have a converter for, and you can actually share the experiment with us, we'd be happy to work on building a converter for you, and then we can actually include the converter in the package so that more people can process those types of experiments, which would be great. Okay, and the last package that I'm going to review here is MSstatsLib. Like I mentioned before, MSstatsLib is our newest package. It was just released last October. Over that time, we've seen about 50 downloads per month. We just released a new version 1.2.1, which includes just some more plotting functionalities. It includes some more vignettes so you can see different types of analysis you could do. It's been developed in collaboration with the Picati Lab. And like I said with PTM, we're still in active development. We don't have as many converters as we do for the other ones. So if you do have some converter you'd like to see and you can share the data with us, please do, and we can build out a converter for it. I think the main converters are Skyline and maybe Progenesis. I'd have to look at the actual package. And it's also available on Bioconductor and GitHub, and the links are in the slides. Okay, so those are the main MSstats packages. There's also MSstats LOBD. I didn't go over it here. Like I said, it's not really integrated in the main workflow, but it's available on Bioconductor if you'd like to use it. Next, I'll go over MSStats Shiny, which we obviously did a lot of work with yesterday, but just to give you an idea kind of of the reason it was developed and when you might actually use it versus some of the code functionalities and when it might be appropriate. So first off, it's available online at the link you can see there, MSstatsShiny.com, or the actual code version is available on GitHub, so you can run it locally. I just want to say that the online version does have limited RAM space, so it's just on a server, so you might not have as much capacity to process large data sets as you do on your local machine. So if you have a small data set, the online version will work fine. But if you have something that's a little bit larger, and you want to run a little bit faster, you probably would want to just run the app on your local machine. Right now, it includes functionality for MSStats and MSstatsTMT with PTM actively in development. And the general goal of this app, it was to increase the usability and reproducibility of the MSStats analysis pipeline. So to give people who maybe aren't as familiar with writing R code the ability to use the packages, and also to be able to have like a simple workflow that different people can use. We can save your parameters and then you can reprocess your data sets later. So it should be really make things easier to actually reproduce as well. Okay. So I wanted to give an overview of kind of when you'd use Shiny versus using the code. So when would you use Shiny? The main ideas when you would use the Shiny application are to use the MSS ecosystem without writing code. So like I said before, maybe you aren't familiar with R or aren't familiar with coding. You can use the Shiny app and run an MSStats analysis the exact same as someone who would be able to write the code. It's really good. Even if you did download it locally, it still is better for experimental data sets that use limited memory. So the app itself, just because it's a UI application and not code, generally takes up some RAM on itself before you even start processing the data set. So it does require you to have kind of an overhead RAM to begin with. And so it really does work better for smaller experiments that aren't going to just take up all the memory on your computer. With that being said, one thing that's included in the Shiny functionality is the ability to produce code for your workflow. So what you could do is take, maybe if you wanted to be able to generate the code that you could use for a larger experiment, you could take a subset of your data set, run it through the Shiny pipeline with all of the parameters that you would like to use. So we have a lot of information on what parameters to choose. Everything that you choose gets put into this code file, and then you can download it. And then all you need to do is replace the CSVs in the code with your larger data sets. And then you can run the exact same thing that you ran in Shiny, just using the code. And you don't have to write the code yourself. You just run it in RStudio in the console, and it will be able to run the larger data set. And one thing that's really cool about that is, like I mentioned with MSStats, we're actively developing the ability to run very large data sets that don't fit into memory with MSStats. So that might not be possible with Shiny, but if you have some really big data set, you could do what I explained, just run a smaller data set, download the code, and then you could use that functionality for the really big data set with the code version. So it gives you a lot of flexibility over how you actually will run the experiment, and producing the code helps with reproducibility. With that being said, when would you want to kind of use the code version instead of the application? So obviously the code version works best when you have to process large data sets that require access to more memory. It doesn't have that RAM overhead that you have to use just to even get into the just to start processing everything. Additionally, one thing I think the code lets you do that MSStats Shiny, the application, doesn't let you do is it increases your flexibility to do analysis and plots outside of MSStats functionalities. So what does that mean? Basically, with Shiny, you need to have a data set from one of our converters or already in MSStats format. You need to be able to put it into the app and then you are limited to the exact functions that MSStats can use to process the data. So if maybe you want to do some like data like exploration on your own that MSStats doesn't include, like some plotting that MSStats doesn't include, like do some histograms or something like that, or do some like change your data set in any way. That all has to be done outside of the application. So if you're comfortable writing R code, the code does give you more flexibility to do all of these different analyses using different packages, different plots that the app doesn't allow you to do because we are constrained to what we actually put into the application. So that's kind of just to give you an idea of what's available between or when you might want to use each of these two, the Shiny or the code version. So hopefully you have a better idea of like what different so hopefully you have a better idea of when you would like to use each of them going forward in your own experiments. Lastly, just to show you where you might want to reach out to us if you do have any questions. We have a Google group, so you can see there. You can post any questions you have on any of the packages, if there's any bugs, or if like you have a question on how you might process a data set or something's not working right. So post there. Matt or I or Olga will be able to respond and give you guidance. Additionally, you could post an issue on the package GitHub, and the links are all in the slides that we've gone over previously. So that's the overview of MSStats ecosystem. If anyone has any questions, I could definitely answer them now.
Thank you so much, Devon. I see there's like a couple of questions related to error messages and things like that. So I think we can just answer them by typing. So then let me go ahead and share my screen, and we will go back to statistical methods, and we will talk about statistical methods specifically implemented in MSstatsTMT. And maybe I will have also, I will touch upon a couple more topics just based on the questions that we received yesterday during the presentation. And after that, we will have a small break, and then Ting will lead the hands-on session on MSstatsTMT and based on Shiny. Okay, so the first thing is there were several questions yesterday asking whether MSstats can fit ANOVA models, and also there were no questions, but usually people ask whether MSstats can fit lemma, and what are the differences between ANOVA and lemma, which are more general methods in bioconductor, and the statistical methods in MSstats. So I will talk about that, and then I will talk about some details related to MSstatsTMT. So first of all, analysis of variance. So ANOVA stands for analysis of variance, and essentially it is an extension of the t-test that we discussed yesterday. Let me just review that. So let's say we have a label-free experiment which has one feature per protein. And so here we have a replicates from the healthy. So this is a set of healthy subjects with these values of protein abundance. These are the subjects with the disease with these values of protein abundance. And so the question we wanted to ask was whether in the underlying population, not just in our subjects, but in the underlying populations, the mean protein abundance in the healthy is same or different from the mean protein abundance in the disease. And so for that, what we did, we specified a null hypothesis saying that, well, we do not know what the population means are, but we'll make an assumption. So this assumption is that there is no regulation, so there is no change in the population level in protein abundance between healthy and disease. The alternative is that there is a change. And so to find some evidence against the null hypothesis, we built this signal to noise. So the signal is how much difference we actually observe from our data. And the noise is how much uncertainty we have associated with the two means. And so you remember that In the simple case, the signal was just the means in the two groups, and the noise was the standard error associated with those means. Remember, we talked about the central limit theorem, right? We talked about those S1 and S2, their variances in the population. So we have to divide by the sample size to reflect the uncertainty in the means. So this is what we discussed yesterday. Now, the problem, and in fact, there was a question about that also yesterday in one of the Q&A, what happens if we have low intensity data, or what happens when we have noisy data? And so usually what happens is two things. So either one of the two things. So either our data are very noisy. So if we have very broad spread of protein abundances, either because the protein is just highly variable naturally from one individual to another, or because our measurement technology adds a lot of noise, then the signal can be very small compared to the noise, and we do not have enough sensitivity. Even if there is an actual regulation, the noise in our data can be so high that we will not be able to detect the signal. So this is one thing that can happen. And the other thing is when we have a small sample size, then occasionally those signals tend to be exaggerated, because let's say if we only had 2 replicates in this group, we have replicate here, in the other group we have replicate here. So it appears that the signal is much larger than it should be, just because we haven't sampled enough of the population. So these are the problems, especially when the sample size is small. And so there are multiple techniques to handle this. Well, one technique obviously is increase the sample size, which is the easiest one, right? But there are also other techniques we can use to add stability to our signal to noise estimation. And analysis of variance is essentially an extension of this type of approach, which focuses on adding more groups to our experiment. And so here is an example. So this was a collaboration that we had a long time ago with clinicians who study cardiovascular disease. So now instead of 2 groups, we actually have 5 groups. Each group has its own population mean of protein abundance. There is absolutely no reason why every group has to have the same mean protein abundance in the population. However, what we can assume is that the variability underlying each group in the population is the same. So the spread around the mean is the same in each population. If we can assume that, then we can learn the amount of the variation in each population from all the groups. Even if we're interested in comparing these 2 groups, we can leverage the information about the variability from the other groups to essentially gain stability for our estimates of noise. So this is a technique related to the experimental design. So of course, first thing, just increasing the sample size is definitely a good idea, is definitely helpful. But another way to add stability is actually add more conditions. Now this comes with the price of the assumption that the variation is the same in every group. If it is not the same, then we need to do something more complex potentially. And so the question was whether MSstats or MSstatsTMT can feed this type of models. The answer is yes, but only when it is appropriate for the experimental design. So we will need to see whether the experiment contains more than 2 groups. We also need to see whether all of these observations in each group can be assumed independent. If they are not, such as in experiments with TMT, as we will discuss, then we need to do something else. So yes, we can fit ANOVA models, but only for the experimental designs when this is actually appropriate. And the good news is that MSstats does a lot of parsing in the backend to figure out which model is actually appropriate for each experimental design. But this is the use of the analysis of variance potential. So, and this is just some details on that, right? So what we can, so we can, when I say that we learn the variance from each group, what we do, we actually say, okay, let us combine the estimate of variability, assume that it's the same variability in every group, and we have this pooled average of variation, rather than having these 2 separate estimates of variation. Okay, so this was the ANOVA. Now, the second thing that we can do is to add empirical Bayes moderation. So let me just review here a little bit, right? So if we assume the same variance between the groups, we just have a combined estimate of variance, and we have a combined estimate of sample size, and now we just have one. So using additional stability for the noise, so for the denominator that we have here, can also be done with additional procedures. And this is what Limma is actually implementing. So analysis of variance says, okay, let me borrow the information across multiple groups within the same protein. Limma now goes one step further and says, let me also borrow the information on the variance, not just within protein, but across multiple proteins. So now it goes one step further in that. So first of all, it will assume, just like analysis of variance assumes, that we have a common variance between all the conditions for each protein. So now this is how we have our signal to noise ratio. And same problem as before, if the sample size is small, this standard deviation in each condition, so this spread here, may be unstable just because we don't have enough data. And so Limma says, well, okay, but we have more than one protein. We have potentially thousands of proteins in our experiment. Can we somehow borrow the information across them? Well, one very kind of drastic way of borrowing the information is to say every single protein has the same variance. And this is way too restrictive because, of course, we know that every protein is not regulated in an identical manner. So some proteins are more variable, other proteins are less variable. So assuming that every protein has the same variance is really, really a strong assumption. What we can do, we can have somewhat of a middle ground, right? So we can assume that, okay, these variances of individual proteins, they all come from some kind of, common pattern, which is not a constant, but some kind of common pattern. And so technically, and this was developed quite a while ago for gene expression microarrays, but it's now also popular for proteomics. So what we do technically, just for technical reasons, we do not consider the variance of the proteins, but 1 over the variance. And we essentially plot a histogram of this quantities 1 over the variance over all proteins that we have, right? So we have many proteins, we assume common variance between the two groups, right? So we calculate this variance like this. And now we make a histogram of these variances over all the proteins. And then we can fit a particular distribution. This distribution is called inverse chi-square. And so essentially fitting a curve to this histogram. And this curve has 2 parameters. There is D0 and S0. And this S0, you can think of this as the average or consensus variance over all the proteins. So this would be the variance which we would use if we wanted to assume that every protein has the same variance in the data set. And then D0, you can think of this as a weight, is how common it is for all the proteins to have the same variance versus not. In particular, so if this histogram is very tight, so if it doesn't vary very much, then D0 will be very large. If the histogram is very broad, it means that from one protein to another, there is a lot of kind of differences in the variance. Then this D0 will be small. And so now there's actually quite a bit of theoretical argument, which essentially boils down to the following. Instead of using a protein-specific variance in this noise part, we use a weighted average where we replace the variance with a weighted combination of protein-specific variance and the common constant variance between all the proteins. So before, each protein has its own variance, and it may be unstable if the sample size is small. Now this variance is stabilized because we have a weighted average between something which is calculated from the entire data set and something which is calculated only from the protein. How big the weights are, well, this is determined by this curve fit and by how flexible or widely spread this curve is. So to summarize, what Limma is doing is that it improves the stability of variance for comparisons such as two-group comparison or analysis of variance comparison. It is a good idea, it is helpful. What lemma does not do, it does not account essentially for differences in the signal. So it still assumes that all of the points that we have are independent, and it does not account for more complex structures of experimental design, which have some kind of similarities between observations, such as time course data, or paired designs, or repeated measurements, or mixtures if we have a multiplex experiment. And so lemma is really helping with the stabilization, but for many experiments in proteomics, especially in multiplex proteomics, the signal part of lemma is actually not quite adapted to what we would need. And so what MSstats is doing, it is kind of using the same ideas of stabilizing the variance denominator, but it models separately the signal specifically to the needs of quantitative proteomic experiments. And because the whole point is to improve the stability of the variance, if in fact your experiment has a large sample size, this type of approach, it is called empirical Bayes moderation. So this type of approach makes no difference anymore, because what will happen is that if you have a large sample size, your protein specific variance will dominate, and essentially there is almost no difference between the two approaches. In practice, our experience is that if you have more than 3 replicates, there is really no need for that. This works just fine. But if you have designs which are more complicated than just comparing several groups, then the noise part, the signal part is not quite sufficient in what lemma is doing for the purpose of quantitative proteomics. So we implement this especially for TMT. We, I think, haven't done for the Bayes MSstats because people now tend to have larger sample sizes in their experiments. So it's not as relevant. This is one of the older publications which shows essentially how the variance is stabilized. So this is the volcano plot as we discussed, right? Like log fold change and p-value negative adjusted for multiple testing. So here we see that large fold changes may or may not be statistically significant just because of the instability in the estimation of fold changes, whereas here more systematically large fold changes are associated with small p-values because the transformation is more linear, right, from the fold change to the p-value because the denominator is more similar across all the proteins. Okay, so I hope that Ting and Devon can answer questions as you type them, and if not, I'm happy to answer more questions after that during the break as well. Okay, so now we can talk about MSstatsTMT and what we actually do for a TMT specifically. So unlike label-free experiments where we have, this was an older cartoon, right, that we looked at yesterday, we have essentially one sample per mass spectrometry run, right? So let's say we have a protein digested and we have a particular peak for this protein for the healthy individual, and then in a separate run, we have a peak for the individual with the disease. And so when we attempt to compare the abundances of healthy versus disease, whatever artifacts happen in this run and whatever artifacts happen in this run, they add variation and uncertainty to the things that we are trying to compare. And so this means that we have, so on one hand, the samples are, it's easier to do, the samples do not have to be labeled, there's no complex processing. On the other hand, the price is that we take longer, but also the artifacts, between run artifacts, impact our statistical analysis, impact our ability to detect changes potentially. So an alternative to this is label-based and isobaric labeling. And I hope we all know how it works, right? So that we digest proteins from different samples, and then we attach isobaric labeling tag, like a barcode, to each of the samples, and then we physically mix the samples together. So that now when we have a peak in this M over Z versus retention time space, this peak is actually a mixture from all of these sources that we had. And the quantification occurs at the level of the secondary or tandem mass spectra. So we have reporter ions, which characterize each of the original samples. So now, instead of looking at the intensities of these peaks, we're looking at the intensities of the reporter ions. And we have to summarize all this information as pertained to the protein to ask questions whether, for example, the healthy sample is different from the various disease samples. Now, this is fairly complicated because we are now changing the quantification from MS1 level to MS2 level. And on one hand, we now have multiple MS-MS spectra, and you can imagine that the reporter ions, which are part of a same MS-MS spectrum, they share some similarities. They're more similar to each other than reporter ions which belong to different MS-MS spectra. For example, if an MS-MS spectrum was sampled at close to the apex of the MS1 peak, these intensities will be higher. On the other hand, if the MS-MS spectrum was sampled closer to the tail of the peak, then these intensities will be lower. Simultaneously as a group, right? Just because there was less biological material during this time. So as a group, these reporter ions tend to kind of vary together higher or lower. And so statistically, we have to account for these differences, right? Because they represent the technology and not the biology. At the same time, while MS-MS spectra, they are stochastic, meaning that we really don't quite have the control of how many MS-MS spectra we'll see for each peptide or each protein in every run. And so it is very possible that we will have different peptide ions in different runs. We may have several MS-MS spectra per peptide ion in one run, none in the other run. So there's a lot of uncertainty happens. Traditionally, multiplexing has been used, especially if we have enough reporter ions. These days, we can have quite a few, I think 14 or even 18, so that the entire experiment can be done in one mass spectrometry run. Then we don't need to worry about these issues. MSstatsTMT can do that, but really the strength of MSstatsTMT is working with experiments where we have more replicates or more conditions than what can fit into one multiplex. So we really focus on experiments with multiple mixtures. And this introduces enough statistical complexity that we had to have a separate package for that because it's quite different from label-free experiments. And there are issues such as how we account for different layers of uncertainty, but also how do we design optimal experiments when we do multiplexing. And so that's really what we focused on. So here is how MSstats looks at this data. You have seen a lot of profile plots yesterday for label-free. So here we have a more complicated plot because the experimental structure is more complicated. So here, let me just walk you through. Each panel is a mass spectrometry run. Some of these runs can be technical replicates of the same mixture. Some of these runs can be new mixtures, so that we mix different replicates in different mixtures. Now the vertical lines separate the channels. So these are the labels of the different channels. Y-axis is a log intensity. Again, we always work on the log scale of the peaks. And different colors here correspond to different peptide ions. And you can see a lot of things. So for example, here we have a lot of within peptide variation. Some of this is noise, but hopefully some of this is biology because we will have different biological samples labeled with different tags. And the objective of our study is actually detect these changes on average over all of the data that we have and over all the peptide ions and so on to say, okay, well, these are the groups which are actually regulated. They are changing in abundance between conditions. But we see right away that there is also between mixture variation here. So for example, this is the same peptide ion. And exactly as I was saying, so here we actually have two MS-MS spectra for this peptide ion. Here we have one, and the intensity shifted because we have probably this this mixture. In this mixture, this peptide ion was sampled at a different height of the peak. So this is the type of variation that we have to account for. But of course, a really big challenge for us is the inconsistency of quantification between mixtures. So here, for example, we have an empty channel. So this is a missing channel within a particular peptide ion. This doesn't happen very much, so happens into 1 or 2% of cases. Most likely, if we can sample a PSM, we can sample all the reporter ions within this PSM. However, what can happen very frequently is that the same peptide only appears in a few runs. For example, this red peptide ion is missing from many runs. This blue peptide ion is missing from many runs, and so on. And so this is the biggest challenge for us, because now not only the intensities shift between the mixtures, but also we don't even get to see the same ions, and they have different ionization efficiency and like different properties. So how can we integrate the information across multiple mixtures to still distinguish these artifacts which are due to data acquisition from the actual true biological signals? So this is the objective of MSstatsTMT. And so now let me contrast the label-free versus label-based workflows from a statistical perspective. So here is how we were thinking about label-free experiments, right? So we have multiple runs. Each run corresponds to a subject, or we can even have multiple technical replicate runs corresponding to a subject. Multiple subjects are grouped by condition, and then we can have multiple features with maybe some missing values. So essentially the condition is the top level for the grouping of our subjects. And then subject is the second level and run is the next level. Now, if we work with label-based experiments, things become more complicated because the top level of the grouping now is a mixture. And now within the mixture, we can have many ways to allocate conditions and replicates in one mixture versus in different mixtures. And we just have some kind of typical design patterns which people frequently use that we keep in mind when we think about these problems. So for example, here we have multiple mixtures. Let's say each mixture has multiple technical replicates. So that's already one thing, right? Now, within the mixture, we can have, and within one replicate, we can have multiple conditions. And within one condition, we can, and let's say we have a few conditions, let's say 2 or 3, then we have room to add multiple biological replicates from each condition within each plex, within each mixture. And so now what happens is that multiple biological replicates are similar to each other if they come from the same condition. But also all of these measurements are similar to each other if they come from one mixture and they come from one run. So in statistical language, it creates a correlation of observations which come from one mixture and one run. And we have to account for the correlation. And then again, we have features, but unlike in label-free, where we have missing values here and there, here we have missing values. If we think of a feature as a peptide ion, so then we have missing values much more extensively if a particular peptide ion was not at all observed in the experiment. And so now we have some observations here, but then these observations are missing in other mixtures and so on. So we need to account for all of that. So first of all, summarize the features and then make sure that the features are comparable, and then account for this orange and blue aspects of the experimental design to essentially build again the signal to noise ratio to understand what's the appropriate signal, what's the appropriate noise, and whether yes or no, the signal to noise is higher than as we expect by random chance. So that's really the work. And we will mirror the same workflow as in label-free, but with modifications. So a really important aspect here is experimental design. Yesterday, I talked about the fact that we need to ask ourselves what is the biological question. So whether we want to compare protein abundances between conditions at the population level versus explore protein abundances versus discover biomarkers. Again, here we are interested in comparing protein abundances at the levels of the populations. But also now we have the technological aspects of experimental design. How do we allocate samples to mixtures, right, or to plexes? Is it better to put all conditions in one mix, or is it better to put all replicates of one condition in one mix and another condition in a separate mix? I'll tell you more about that, but let me give you the answer right away. Usually, you want to put representatives of every condition or every time point in one plex. So things that you're interested in comparing should be as close to each other as possible within the same level of this random source, which is a mixture. And then if you have another replicate, you create a second replicate mixture with other biological replicates of that, so that if one mixture is lost, you still have one mixture which has everything, all the conditions in it. So there's more complications, and something that we advocate for strongly is to have one reference channel, which is used for normalization. And in fact, it was already here. I hope you can see on your screens. One of the channels here is called norm. So this is a normalization channel. This channel has some biological material which is added in a constant concentration across all mixtures. Oftentimes it's some kind of pooled sample, and it's a good idea because then we are more sure that this channel has enough biological material that across multiple conditions the protein is abundant enough that this protein will be selected for fragmentation. But also this reference channel will be of a great help for us to make sure that all of these intensities can be corrected so that they are comparable between mass spectrometry runs. Sometimes there is a question whether we need to have more than one reference channel. We tried, we actually had experiments with 2 reference channels. We found that yes, it is helpful, however, not helpful enough to compensate for the loss of a channel that can be used for one biological replicate. So fundamentally, having an extra channel for an extra biological replicate is more useful. So one reference channel is enough. We have seen experiments without reference channels. It is not impossible to analyze them, but then essentially you have to make much more strong assumptions. So you make assumptions that on average, the experiments do not have as many missing values, that the average intensities are the same across runs. Reference channel essentially allows you to freeze you from these extra assumptions. You do not need to make these extra assumptions. And also reference channel allows you to work with unbalanced experiments where different mixtures can vary in number of replicates for each conditions, which happens often, especially if you work with clinical experiments where you cannot control exactly the number of samples. So reference channel really helps. One other thing that we see often with TMT is fractionation. So that also is part of the experimental design, and MSstats can handle that. So that's also one aspect that needs to be included. Okay, so then we have data acquisition, and after that, data processing. So for data processing, as Devan and Mina mentioned previously, we have converters from quite a few tools, including MaxQuant and Proteome Discover and FragPipe, probably most relevant for the course. And the converters don't just format things, but they also do filtering. And so Tim can tell you more details about the filtering step, which is important. So for example, currently, actually all MSstats workflows filter out shared peptides, meaning peptides which belong to multiple proteins. We tend to discard them so that we use only the unique peptides for quantification. There's a threshold for PSM, so for peptide spectrum matches which have many missing reporter ions. It doesn't happen often, but if it happens, we filter them out. If we have multiple peptide ions, so multiple MS spectra with the same PSM, we keep the best. And there are some criteria for the best. This will be intensity, missing values, interference scores, and things like that. Combining fractions. So if a peptide is present in multiple fractions, fairly simple, we just keep the version in the fraction which has maximal mean intensity. So that's data processing. And so now the statistical analysis has 2 parts. One is actually several parts, normalization, summarization, the second normalization, and then modeling and inference. So I will walk you through that. And so again, this is the data structure that we have in mind. So the first step is the spectrum level normalization. So what can happen is that different labels, they have slightly different labeling efficiency, or sometimes things just happen, some kind of artifacts when we mix multiple samples together. And so here, this is how MSstatsTMT visualizes this. So again, each panel is a mass spectrometry run. Each x-axis is the label, and y-axis is the log intensities of all the features in this run. So across all proteins, all peptides, everything. Because it's a DDA experiment, because typically we will have a fairly large number of proteins, we do assume that the median intensity should be constant across the runs. And so the first level of normalization just equalizes the medians across all the runs. This helps so that we kind of benchmark this on different data sets. It does help reduce some of these artifacts due to labeling efficiency and due to some issues with mixing. This is not the strongest normalization, but it helps to some extent. So this is what MSstatsTMT is doing first. This is done across all the proteins. After that, everything we do is done separately for each protein. So this was all proteins together. Now the next step is separately for each protein. As we have seen, right, so in different runs, we will have different peptide ions and different PSM. This run has more PSM, this run has fewer, this run may have some others, so that the intensities between the runs are not comparable. That's okay, because currently we do the summarization separately for each run. If you just think about one run at a time, you can think of this as a label-free experiment, right? So we have different channels corresponding to different replicates. Here we have different features. And so what MSstatsTMT is doing, we summarize this using the same robust procedure as we use in MSstats in label-free experiments. So Tukey median polish. And occasionally you can choose to have imputation if you still have some missing values left, but usually there won't be many missing values left within a run at this stage. And so this would be the summary of this protein across all the features. So the summary of the protein separately in each run across all the features here. These summaries are not comparable between the runs yet because they're based on features with different identity and different intensity, strength, and so on. But now already the values are comparable within the run. So if you have one run, so this really is all you need to do. You would stop here. But the next level of normalization, separately for each protein, now makes sure that the intensities are comparable not only within the run, but also between the runs. And this is where we use the reference channel. So this is an example which actually has two reference channels, just to kind of illustrate what's going on. So this purple circle is the reference channel. The red line is just some kind of average, right? And we see that here the purple points are on the average, here they are below, and here they are above. So what this protein level normalization would do, it would shift the purple reference channels so that they're all equalized. And whatever shift was applied in this run will be also applied to all the endogenous intensities at the same time. And the same thing here. Here it's downshifted, so the shift is applied to all the endogenous intensities. So the assumption is that whatever artifact happened to the reference channels, the same artifacts happen to all the endogenous channels of the same run. And so now that we applied this normalization, now these intensities, they are comparable, not only within the run, but also between the runs. And now we can take advantage of all the full structure of replication and of the full structure of experimental design to distinguish signal and noise. But for that, we need to model what is the signal and what the noise, right? And so these are the challenges for the statistical part. So represent the experimental design, accurately represent the peak intensities, estimate the sources of variation, and do hypothesis testing. So make some statements about population level analysis. So This is the structure of the data as we discussed. And just like for MSstats, this type of designs actually are somewhat known in statistical literature. They are called split plot designs. What is important here is that there is a restriction on randomization. We randomize the replicates within the mixture, then we randomize separately the mixtures, but the features we acquire, they are not randomized, right? They are all acquired simultaneously within each replicate. Because we have restriction for randomization, and this is really important, we have different sources of variation will be considered as noise, depending on what we're interested in comparing. And I will get to that in a minute. But for now, and this is now for those of you who have some statistical background, this will be a more complicated version of this linear mixed effects model that we wrote. So this is a generalization of the ANOVA model that we discussed in the very beginning. In a very special case, when you have one mixture and you have one feature, you can think of this as the ANOVA model, but it generalizes to multiple mixtures and so on. Importantly, it now has multiple sources of randomness. So we acknowledge the fact that the mixture is random, replicate run is random, in the sense that if we have to repeat the experiment, we will not have the same mixture again. It will not be identical, and we will not have the identical run again. And the important insight here is that even though the model is complicated, when we want to compare conditions, which is our question of interest, when we compare conditions, this comparison only depends on the summary of the features. It does not depend on the individual features. So we can do something similar to MSstats, where first, after normalizing and summarizing things, we kind of work on the summarized version of the data. And this allows us to specify a model which is simpler. Now, I'm not saying it's a simple model, but it's definitely simpler than what kind of original statistical version would look like. And the advantage of this approach now is that we can, again, generalize this to experiments which compare more than two groups, compare multiple groups. And of course, oftentimes we do TMT because we have even more complicated designs, such as time course, such as pair designs, or some hybrid designs have both multiple conditions and time points. And so these models allow us to actually fully express the complexity of the designs and capture the right source of variation to build the right signal to noise ratio. And just to kind of add to this, so remember lemma, right? Because oftentimes we have a few mixtures and variances can be unstable. Some of these noises that we estimate, we can actually use the same empirical Bayes moderation where we borrow the information across all the proteins. So there is that aspect. So it is similar in lemma in that we stabilize the variance. It is different from lemma because we account for more complex structure of experiments when we start having TMT labeling. So just a little bit on the results, and Ting will tell you more about that. So what is important for us is several things. Well, of course, the performance is important, but what is also important is the number of testable proteins. So how many proteins can we actually analyze with this approach? And it turns out that because, lemma, but also sometimes people use HR or some commercial tools for this type of analysis, it turns out that when standard methods were developed for other purposes, like for gene expression, and we just borrow them for proteomics, they don't quite account for the patterns we have in proteomics. And it turns out that some patterns of missing values or some patterns of values that are absent, they cannot be handled with these implementations. In particular, HR. Well, first of all, HR is not even designed for quantitative intensities in the first place, but it's also not designed for certain patterns of missing values. And so this is a problem. So it reduces the number of proteins that can be tested in the first place. And then there are others. So for example, lemma in particular, in many cases, this empirical Bayes assumption is too strong. And so the variance component is too small. And so there are too many false positives. And proteome discover is kind of okay overall. But if we look at how we kind of actually estimate the false discovery rate, so how often we are wrong among the proteins which we claim are differentially abundant, or often we're correct. So we can kind of see that the MSstatsTMT balances well the true positives and the false positives. So we'll tell you more about that. So I hope that Ting and Devon can answer some questions if you have. And again, I'm happy after I am done to answer more questions about this in writing. And so now the last part I just wanted to share with you is essentially a little bit more about complex designs. So if we have one mixture and two groups, and they're all in one TMT plex, it's really not that complicated. And when you don't need to worry about statistical sophistication. So we're really interested in experiments which are larger scale and have more complexity. And so the first aspect here is how to allocate the resources for future experiments. And remember, I was telling you yesterday that we often think in terms of sample size, how different experiments can be more or less efficient in terms of requiring more or less sample size. So this is the type of considerations that we have. Not all of this is implemented in MSstats because this type of considerations need to happen before collecting the data. But let me tell you how people think about that, how statisticians think about this in general. So if we have, and this is a simplification, this is from an older paper that I wrote with Anne Oberg a long time ago, it turns out to be very relevant currently. So if we, let's say we have 5 disease groups, and let's say we also have a multiplex experiment with 5 labels, just so that I can draw the picture easily, right? So then things are kind of clear. So we have one block, so one mixture, right? One plex, which will have one biological replicate from each disease group. With more replicates, so then we have another mixture, which will have one biological replicate from each group, and so on. So this is called a randomized complete block design. It is randomized because we randomly select biological replicates in each mixture. It's complete because each disease group is represented in each mixture, in each block. So these designs are very effective. Well, one, because if you lose a mixture, if your run fails, you still have other runs which are present. This is one thing, but the other thing is you can show when we talk about the signal to noise ratios that it minimizes the noise. And so it will also lead to the smallest number of replicates compared to some other allocations. So this is a very effective design. And I think it's probably not a news to most people here. We know that if we have multiple conditions, we want to allocate them all in the same mixture. But now what happens if we have only 4 labels? So what happens, and again, just for the sake of the argument, if we have more groups than labels, we cannot fit every group in one mixture. So then we have many ways of allocating groups to the mixtures. Well, you will probably say, hey, just let's have fewer groups. That's an option, right? But sometimes we really need to have more groups, and we cannot have enough labels. So there is a solution for that. And a solution for that is called a balanced incomplete block design. It is incomplete because now we cannot have every group presented in every plex. However, it is balanced because we can do this in a manner that is quite systematic. So you see that, for example, group 5 is only missing once from the mixture, and it's present in all the other mixtures. Group 4 is only missing once from another mixture, and it's present in every other mixture. Group 3 is only missing once. So essentially, every block is having every disease group missing once, and every disease group is missing once in every block. So this type of balanced design can be shown to be most effective, meaning, again, it minimizes the noise in our signal-to-noise ratio as compared to some other non-systematic allocations. And of course, we can do this even more extreme. What if we only have 2 labels? Imagine you're doing SILAC, for example, right? So then if we have multiple groups and we have multiple labels, we can show that the same kind of balanced incomplete idea can be extended to this pattern. Now, the problem is that we have now this pattern has to be has to require more runs. For example, here, we need to actually have 5 mass spectrometry runs to make sure things are balanced. Here, we don't care. We can have 2, 3, 4 blocks, doesn't matter. Here, we really have to have 5 blocks present. Otherwise, things become unbalanced. So if we lose one run, it's more of a problem. Same thing here, we need to have 10 blocks in this case, so 10 runs to make sure that this balanced pattern is preserved. But again, if we can do that, then it means that our noise will be minimized compared to some other allocations of resources. In particular, the reference sample, for example, in SciLac, you can imagine spiking reference peptides for every endogenous run. So this is actually less effective from the statistical perspective than doing this type of balanced design. And so this is how statisticians will think about that. So remember this noise, the denominator in the signal to noise ratio. For all of these designs, we can actually calculate what the noise will be analytically. And so here is what happens if we can fit every condition in every block. Here's what happens if we can't fit it. So there's this coefficient, which is greater than 1. So it is larger than this, this part. However, we can calculate what it is. And so you can actually do the same type of sample size here. So this is what happens if you have a complete design, so enough labels. And these are the designs with 4 labels versus 2 labels. So we see that 4 labels is more efficient. It will minimize the noise as compared to 2 labels. So this is not implemented in MSstats. This is something which requires statistical expertise that is specific to your experiment. But this is to say that there are ways to allocate the experiments in a more smart way. And so I guess the message here is that 1, talk to a statistician when designing these experiments. And 2, if you do have such an experiment, MSstats actually can analyze it. So MSstats can work with this incomplete designs or unbalanced designs and things like that. So there's really no limitation for MSstatsTMT in terms of how the samples are allocated to mixtures. So now I would like to just very briefly mention the designs with multiple sources of variation, because that's also what happens very often. And let me again highlight Ting. This was Ting's work as part of her PhD. So this is a fairly technical statistical work, but I will try to give you the intuition rather than technical details. So here is a simplified cartoon. So let's say we have 6 channels in a mixture. And so this will be an experiment with 2 mixtures. This is mixture number 1. This is mixture number 2. So we have 12 samples from 12 distinct people. Some of them are healthy and other disease. So condition 1 and condition 2. The experiment I discussed so far means that we have in one mixture 3 biological replicates from one condition, 3 biological replicates from another condition. This is mixture number 1. And then mixture number 2 will have 3 biological replicates from one condition and 3 biological replicates from another condition again. So that's what we discussed so far. So if you have only one mixture, you can do ANOVA here, there's no problem. If you have 2 mixtures, you need to account for the fact that these measurements are more similar to each other than these measurements because they came from the same mixture, they share some artifacts. So this is what we can do. But oftentimes we are actually interested in measuring the same subject multiple times. Imagine, for example, that you have a person before treatment and after treatment, or you have a time course series, I don't know, some cell lines and they're stressed somehow, and then you measure the response to stress in time. So then we have yet another similarity or another correlation between the measurements, because measurements which come from the same person, they tend to be more similar to each other than measurements that come from different people. And so here is one example. So again, 12 samples, the same as before, but now in mixture 1, we have 3 replicates from 3 time points of one person. And here we have 3 replicates at 3 time points from the second person. So this is mixture 1 and this is mixture 2. So now we have 2 layers of correlation, or 2 layers of randomness. One is the person, because all of these measurements come from one person. and this is another person, all the measurements will be similar to each other. But then the second layer of correlation comes from the mixture, because they were multiplexed together and they share some data acquisition artifacts, and those guys share some other data acquisition artifacts. And so this is where statistical modeling is important, because this can be analyzed with ANOVA. This cannot be analyzed with ANOVA. Here you need to have more complicated models which have multiple sources of variation. So technically in statistics, it means multiple variance components that need to be quantified from our data. And that's where things become fairly complicated. So I will not really go into the details of what these models look like. I'm happy to do this after the presentation, but I will just do two things just to provide you the intuition. One is, so maybe just one more thing I will say before that. If we want to compare time points, I think it's probably intuitive that comparing time points within the same person will give us less noise than comparing conditions between people, right? Because here we just want to see how things evolve in them, whereas here we want to see how things change between different people. Here the person is similar to itself, things just change in time. Here we have a difference due to conditions, but also difference due to different people. So this will be noisier comparison than this comparison. And if we model this correctly, then we will make sure that these comparisons are more sensitive. They have less noise in the denominator than these comparisons. And so now, this is just some sense of how different models think about this. So this is for MSstatsTMT, including this empirical Bayes moderation. So this is the denominator in the signal to noise ratio when we compare times. This is the denominator when we compare subjects. And you see that, and each point is a protein. And so we see that most proteins, comparisons between subjects are more noisy than comparisons in subjects. And it's quite intuitive. But now if you think about, so these two, they're equivalent to ANOVA and t-test. These two, they're lemma with different terms. It's interesting what happens. So these models effectively, they do not have the ability to distinguish the noise within subject and the noise between subjects. And what they do, they just apply some linear transformations essentially. So this is not the right way to distinguish these measurements. And so we think that this is really where the main advantage of MSstatsTMT is, where we have multiple mixtures, but also multiple layers of comparisons, for example, between subjects, within subjects, or between subjects. And here, just some, again, technical argument. Let me maybe start with this first. So this is more for those who are familiar with statistical methods. So if you look at comparisons between conditions versus comparisons between the times, if you look, well, the estimates of variability are different, but if you look specifically at how much data went into these comparisons in the degrees of freedom, we see that all the methods except MSstatsTMT, they use the same degrees of freedom. It's another indication that the within subject and between subject variation is not distinguished appropriately. And this is just to show that when the features are more variable, we will of course have larger p-values, and when the features are more consistent, we have smaller p-values. Okay, so as I said, it was somewhat technical, but I'm happy to clarify, and I'm sure Ting can also clarify a lot of things here. So very last thing, and this is something that we would like to share with you, and tomorrow there will be even more discussion on this topic, but I would like to give a preview already, is how can we benefit from the data which have been collected previously and analyzed previously. This effort in terms of data availability and reanalysis came out of the many method development efforts in our lab, because as we develop statistical methods, of course, we need to test them on many data sets. And data availability is a very big issue for us, because we want to work with data sets acquired on different workflows, on different instruments from different labs, to make sure that things are really stable and generalizable. And while doing that, we realized that being able to share and reuse data is really key for our community. And of course, we have repositories such as Pride and Massive, which store a lot of the information about the spectra. But what is missing is the annotations of the samples and the information required to actually do statistical analysis. So results of peak quantifications, results of normalizations, and so on. And tomorrow we'll hear a lot about that. There is a lot of efforts in the community to enable that. And so we have developed, in collaboration with Massive, we have developed one such way to store and reanalyze data. So this is called massive.quant, which is an extension to Massive. So this is work by Mina, so she will be talking about this tomorrow. But essentially, Massive Quant allows us to store documented analysis of quantitative mass spectrometry-based experiments, not necessarily proteomics. So we now increasingly have mass spectrometry imaging and other data sets also in Massive Quant. The specific feature of Massive Quant is that it doesn't really prescribe any particular way to analyze the data. So even though we contributed to developing it, it's not at all by no means MSstat-centric or anything like that. It is also not dependent on any particular data processing software. So essentially, you can submit your data, you can submit the results of data processing, you can submit the results of quantification, you can submit results of statistical analysis in with whatever tool you used. And the idea is that people can start at any intermediate step of data analysis. And for example, if you want to compare different statistical methods, you have the results of quantitative analysis stored and available for you. So this is very briefly the structure of the repository. So it has very general kind of bins. First is, okay, how the data were acquired. So the raw files, then the identification, so output of identification of spectra, quantification, how the peaks are found and quantified, and then statistical analysis. So you can use any of the tools. If you have executable code, you submit the data and the code so that it's entirely reproducible. If it's something that is point and click, such as commercial tool, you submit the input and the output and the tool version. And then the idea is that you take a data set and you can analyze it with some tool, and then you can analyze it with some other tool, and you can have any other package that you want. And these data sets have unique identifiers that you can track down all the steps, and you can start essentially with in any place. You can replace one identification tool with another. You can replace one statistical method with another. And it helps us understand, again, how much our choices of data processing and of statistical analysis impact the underlying biological conclusions. Because again, as I was saying yesterday, the mission of our lab is to make sure that we detect as much biology as possible, and we're as little biased by the artifacts of data processing as possible. So that's all I had to share for today. So let me once again thank everybody who contributed to this work. I would like to highlight Ting, who did a lot of work on the TMT part of things, Devon, who has been collaborating with Ting on the Shiny part that you will see in a minute. Of course, Mina, who has been behind the method developments, and Mateusz, who is a lot behind the general implementation and scalability. And also I would like to thank in particular the Genentech, who has been supporting quite a bit of this work. So I will stop sharing screen now. So I am happy to answer questions if you have any in writing, but I think it is a good time to take a break. So don't go too far away. Let's come back quarter to the hour. So 12:45 Eastern or quarter to the hour, wherever you live. And we will look at the analysis of TMT experiments now with the hands-on, with the Shiny, but also if you have questions about the code or error messages or anything like that, it's a really good time for that. So don't go too far away, and we will be back at 12:45.